in, and someone's like, a grouse, and so everyone like gets up and like uses the little telescopes that they have in there, and then we're all like looking for it.
Oh my gosh.
So funny.
The videos.
I spent like half an hour last night trying to upload it.
Oh, I had a hard time this, yeah, I did it, I uploaded it this morning.
What video?
Oh, not here, not for you, it's for our.
Oh, and the musical I watched, they didn't have a bad job. It was like, what? So like, it was good, but the singing was kind of bad.
You guys had to sing it in video. Oh,
No,
no.
it was fresh. We were gonna watch stuff.
Like watch stuff and then like make a video about how it relates to our identity and culture and how to.
I watched the Hairspray. It doesn't sound like a movie.
What?
In the musical. Oh, I only watched like 30 minutes of it. The girl was kind of bad singing. So. I just watched some music.
I just noticed that the clocks are actually set for the right time. That may be like the first time in 33 years I've been here. After the clocks change, where the clocks are in reset. Well, good afternoon, everybody. I hope everybody had a good weekend. Whitney and Emily are jet lagged, or at least will be, since they traveled from the East Coast last night and early this morning. And probably slept a little, but being youngsters, their bodies are designed to not sleep very much. So, no. When I was your age, I didn't sleep very much.
You still don't sleep very much.
Oh, that's true too. Because of that time shifted, I don't sleep. So, anyway. I am currently starting to grade the metabolism lab report. I know that you turned that in kind of a long time ago, but I felt I needed to grade the proposals first, so I got those done. It was done on Saturday, and so tomorrow, if your proposal needs revision, that's what you should spend your lab time on. Tomorrow. If it doesn't, then you should start plotting out your projects. You can request equipment tomorrow if you would like to do that. And even for those of you who have approved projects, it might be good just to run your methods through with me one last time before you go out and do it, so I can help you catch any potential issues that you might have. So even for those projects that I've approved, there are, I made lots of suggestions on them, so you should pay attention to those. Okay. So anyway, okay, so exams. Please be patient with me on those because I got a lot of grading to do, and Emily and Whitney have a couple things to catch up on, being gone all last week, and so Emily, you look tired a little bit. Is the day starting to catch up with you a little bit?
A little bit.
Yeah.
Yeah.
Okay. Well, I will rivet you today so that you can stay awake. Anyway, so I will get to them as quickly as I can, so I appreciate the patience on that. But I will probably work through the metabolism lab reports first, and then I'm probably going to jump to my PHYS exams and grade those, and then I'll come back to this class and work through them as quickly as I can. Do you guys have any questions for me before we get started? We're about to start case study 6. My guess is that is that we will get to lightning presentations probably on Friday. That's my guess. Yes. Oh, no, we won't. No, you know what? We won't do lightning presentations on Friday because I won't be here. I will be at the Murdoch meeting, goodness gracious. I will decide what I'm going to do with class on Friday, but we probably won't do lightning presentations. So let's plan lightning presentations for Monday. Okay. I am a bit ahead anyway. I think the reason that I'm ahead is because this class is a lot smaller than the last time I taught it, and I made the decision to do lightning presentations in groups. Had I to do it over again, I might not do that. I don't know, but it is what it is. And so it's conceivable that I will end my already prepared material a little bit earlier than planned, and if I do that, I may drop another case study in. We'll see how it goes. But anyway, so I'll decide what I'm going to do with Friday later in the week. But I won't be here Friday because I have to go to work. Okay. Thanks for reminding me about that. Anything else I should know that I don't remember? That happens a lot more lately. Take that as a no. You ready to get started? Talk about crabs and parasites? Yep, let's do it. Okie dokie. So this study assessing the effects of trematode infection on invasive green crabs in eastern North America. I know that you have been waiting with bated breath to hear about this. So the basic premise of this study is the authors were interested, and this is the big picture sort of thing, in whether or not natural controls develop through parasitism in an invasive species. So this European green crab is a crab that has kind of hitched a ride, usually on boats, and made its way to like 5 continents. So it's pretty widespread invasive species. And the problem with invasive species, and we saw a little bit of this when we were looking at the ants a little bit earlier, is that you remove them from a circumstance in which they have a natural control. So predators, disease, whatever, all right? And so the authors are interested in trying to understand if, in fact, a natural control might develop for these European green crabs. And specifically what they're interested in is a natural control through a parasite. So, shipping methods. Ships have been responsible for spreading a whole bunch of invasive species. A lot of them invertebrate, but some vertebrate species as well, rodents in particular. And they can impact ecosystems just about anywhere they go, because you dump them into an ecosystem, the ecosystem is not designed for these invasive species. It's they can really disrupt the trophic structure that we see in an ecosystem. Because, as I just mentioned, they have no natural predators, the diseases are not really designed for these invasive critters. They can utilize resources perhaps better than native species can, so it can really disrupt an ecosystem. So We refer to these types of species as non-indigenous species, or NIS. And as I mentioned, they wreak havoc because they're released from all of the natural controls. And we've mentioned predators and parasites, but competitors is also another component of this. And this is usually because they're better competitors than anything else that's in an ecosystem that is indigenous. And the reason why they're often not as good is because the niche of indigenous species has been crafted in order to allow the ecosystem to work well. So if you think back to our ants, where we had an invasive ant species that was really good at utilizing a whole bunch of resources there, as opposed to the dominant native species, which wasn't quite as good, but it was good enough that it allowed it to be dominant, but also allowed it to allow the natural biodiversity that was part of keeping the habitat healthy to sustain itself. But the invasive species would dampen down biodiversity because it was too good at utilizing resources. Okay. And so when you get groups of organisms that co-evolve together, you usually see a group of organisms that achieve that sort of a balance. But when you just dump an invasive species in there, that doesn't happen any longer. Okay. So when you take an invasive species out of its natural habitat, what you find is that their parasite loads decrease rather dramatically. And again, this is because the parasites that they find in their new environments, wherever they get dumped off, are not designed to infect these invasive species. That's not what they have adapted to do. And so these invasive species can get another benefit by being invasive because they survive better, because they don't have to experience the same level of disease. They reproduce better. So survivorship and improvements in survivorship and reproduction mean your fitness goes up. And then they just tend to be healthy, which also it can benefit your fitness. Okay, now what the authors were curious about is what happens if after an invasive species has been in a new environment for a period of time, what is the likelihood that they may pick up a parasite that's more of a generalist? In other words, has the capacity to infect a broad range of organisms. Could something like that infect an invasive species and then begin to serve as a natural control for that invasive species in order to keep it from getting out of control? Okay, the most common parasites that kind of serve this sort of a role are Digenean trematodes. So these are flatworms, and you all remember what digenes are from our first couple weeks in the class, right? So these are flatworms that have an oral and a ventral sucker. They have multiple life cycle stages and typically involve more than one host. Okay. And so the life cycles are actually rather complex. In our second lab exercise in this class, you guys did some drawing of the life cycle of an example of a digene. Okay, it was a liver fluke. So this is another one of those that has a fairly complex life cycle, right? So these flatworms, these trematodes, have their strongest physiological and behavioral effect on intermediate hosts. So not the final host, which is usually a vertebrate, okay? But one of their intermediate hosts. And the reason for this is because if they can alter the physiology or behavior of these intermediate hosts in such a way that improves the likelihood that they are going to put that larval stage, that life cycle stage, in a position to move on to the next life cycle stage, then all the better. And so a lot of times these intermediate stages, these life cycle stages of the flatworm will cause the host, the intermediate host, to behave somehow differently that exposes it more to maybe getting eaten. So that the next host in line, it can infect the next host in line and develop into its next stage. So physiological impacts that a life cycle stage of a flatworm can have on an intermediate host. It includes things like castration. That sounds painful. Impaired reproductive function. I think castration actually would fall into that category. Okay. Tissue damage and a variety of tissues. And that's because often these intermediate stages will insist in tissues. So that can easily damage it. Immune activation. We'll show some examples of that. Diversion of energy resources. So in other words, the host would preferentially like to invest energy in one thing, but the infection requires it to invest energy someplace else. Okay. And then organ dysfunction, which may also kind of fit in with tissue damage. Okay, so those are some physiological things that a life cycle stage of a flatworm can do to a host. Behavioral things are a little more obvious. Change in host foraging. So in other words, maybe they will initiate a behavioral change that causes a host to forage in the sun rather than the shade, because the likelihood of that host getting eaten is going to be higher in the sun than it is in the shade. Or use of shelter. There was an example I used back in the days when I taught ecology about a flatworm who infected a snail. I think it was a snail that that got it to forage on top of the leaves instead of underneath the leaves where it was more exposed, and then starlings would come along and eat it. Okay. So that was a behavioral shift. So predator avoidance, and then conspicuousness, just encouraging it not to stay hidden. So all of those things can promote advancement of the parasite's life cycle. So in this study, the focus was on parasitism on the European green crab. And because it is so common worldwide, they're just really interested in knowing is, can this thing ever be controlled? Questions at this point? Yes?
I guess like further with the study to like look, sorry, where's that part? Like look for biological control methods like to implement, or is it like?
No, I think that what they were interested in is understanding potential mechanisms, natural mechanisms that could develop into a natural control.
Which then you don't have to worry about like introducing another thing into the
Yeah.
environment too.
Now that's not to say that if something like this proved to be effective, that if an invasive species was a problem someplace, that they wouldn't introduce a parasite that could control them. It's not to say that they wouldn't do that,
Yeah.
but that is not the intent of the authors here. It's more of a case is that these green crabs do get infected by a particular parasite, and they're wondering how effective that parasite's going to be at controlling the green crab. Okay, so the green crabs in their natural environment, which would be in Europe because it's called the European green crab, has quite a few natural parasites, but they escape about 70% of those when they are introduced into a new recipient habitat. So clearly their parasite load is going to be down. There is one parasite that appears to impact this green crab pretty much everywhere it's found, and it's this Microphallus similis, and similis here, which is a flatworm. It's a diagenetic trematode. And they were, like I said, they were curious to see how much of an effect it had on this organism. So the M. similis is a trematode, has a 3-host life cycle. So it's a fairly complex life cycle. And the crab is the middle host in this life cycle. And the particular stage that infects the crab is called the metacercaria. And we'll go over what that is here in a moment. And it's a stage that will go in and it will encyst in the tissues of the green crab. So here is a life cycle of a digene. It's very similar to M. similis, but it's not exactly the same because M. similis, I'll show you the M. similis life cycle in the next slide. The final host of the M. similis life cycle is actually a bird. But it starts when eggs are added to the environment. This is usually through fecal matter. That's where the eggs end up. And then when the eggs get into water, they develop into a stage, it's called the merocydium. You should remember these names from art lab number 2. The merocydium is ciliated and it swims. And then it will make its way to the first intermediate host, which is a snail. And then when it gets inside the snail, it actually starts a series of life cycle stages that actually represent an amplification of reproduction. It's an asexual form of reproduction. So the merocydium will develop into sporocysts. The sporocyst will produce a number of the next larval stage, which is called the rhedia. And the rhedia will then internally develop a number of the next larval stage, which is called the cercaria. The cercaria then will bore out of the snail and become free living. It's a swimmer. And then it will bore into, in the case of our parasite, a crab. The crab is not involved in all life cycles of flatworms, but in this particular one it is. And then it will encyst in an organ called the hepatopancreas, and we'll talk about what that is in a minute, and I believe also in the gonads of this crab. And then what happens is the final or definitive host will eat the crab. And as long as they are eating the portions of the crab that have the metacercaria encysted in them, then the definitive host will become infected and the metacercaria will develop into the adults. It will result in an adult. And then the adults will lay eggs and start the process all over again. So you can see there's a lot going on in this life cycle. All right. So here is the specific life cycle of M. cereus. So the definitive host here is a gull. And it will poop out the eggs. The eggs will then become the merocydia. Whoop, I'm going the wrong direction. Here are the eggs. The merocydia then will enter the snail. We'll go through all of its stages there. And when the cercaria develops, it will leave the snail, become free living, bore into the crab. And then the crab will develop the metacercaria, and then the gull will eat the crab. That's kind of the way that it works. Okay. So, questions? We all kind of get the life cycle here? All right, so understanding how an infection by M. similis will impact these green crabs is important because they're really interested in knowing is there anything going to control these green crabs? Because these green crabs get in and start competing with other shore crabs. And they grow bigger, they tend to be healthier, and so they just do better in the environments. And as I mentioned before, it could really disrupt ecosystems. So the authors are going to use laboratory and field experiments to look to see whether or not M. similis actually impacts either the physiology or the behavior of the green crab, to see if this parasite actually can impact the fitness of this invasive species. So specific things that the authors are going to look at. They're going to look at just mass and energy. That includes the contents of the hepatopancreas and gonads. They will look at immune activation, and then they will look at things that are associated with foraging and predator avoidance, some behavior that's associated with that. We'll look at those. We will look at those data probably on Wednesday. We'll see. All right. Since most of you probably have no idea where a hepatopancreas is located, I thought I would show you pictures. So here actually is a crab. This is actually a mitten crab. And the hepatopancreas is located right there. So this is the head, the hepatopancreas, and also gonads. I believe these are the testes right here. Ovaries would kind of extend up in this direction in the crab. Here's another organism. It's not a crab, but it's a shrimp. And I put this one in here just so you could see, a live organism that has its hepatopancreas, and that's that structure right there. Okay. So that's your anatomical orientation to where these things are located when we start talking about them. This here is an electron micrograph that shows a hepatopancreas. We'll look at this panel right here because it's the best, I think. So you got a lot of normal hepatopancreas tissue right here, and then scattered you have these cysts that are the metasacharia. And so infections can be heavy, they can be light, depending on how much exposure the crabs have to the parasite. All right. So what does a hepatopancreas do? It's a digestive organ. And so its role in metabolism is it participates in digestion and absorption of food. It will actively secrete and synthesize digestive enzymes. So it literally is, functions as a digestive organ. And then it also plays a key role in carbohydrate metabolism. So those are the key things that the hepatopancreas does. So the authors are going to use a couple of pretty simple but standard indices for evaluating the health of the hepatopancreas. And maybe health is too strong of a word. Maybe state of the hepatopancreas is a better term for that. And basically, they're going to use what's called the hepatosomatic index, or some people just call it the hepatosomatic index. Well, now, they leave the S out of it. So hepatosomatic index, some people just leave the S out of it, just call it HI, and you'll see an image that does that. Or the gonadosomatic index, which is GSI or just GI. Okay? And what these do is they basically are measures of the proportion of body mass that these organs represent. And so as they get bigger or smaller, it can tell you something about energy storage or reproduction. And I'll give you some examples. Okay. So what the authors will use this index for is to get some idea as to how energy is being mobilized. So just in a general sense, if the hepatopancreas is getting bigger or getting smaller, then that is an indicator that it is either storing energy or it is using energy for something, transferring energy to something else. All right, here's how it's calculated. It's a real tough calculation. The dry mass of the hepatopancreas or gonad minus the metasaccharial mass, so you take the parasites out, divided by dried total body mass. That's a toughie. You got to have at least calc 3 to make that calculation. Oh, come on. I should get at least a chuckle. All right, there you go. So that's how you calculate HSI or GSI. So here's an example from a shrimp. Okay, this graph here is for males, and they plot HSI as a function of GSI, only they're labeling it as HI and GI here. And what you see is there's a negative correlation between the two. So as the HSI gets smaller, the GSI gets bigger. Any idea what that might mean? Go ahead, Emma.
I was just trying, my brain's on like 2% speed. It's like where the production output go, or like where the allocation of energy.
So where's the allocation of energy going?
To production.
Okay, so specifically in this case, the gonads.
Yeah.
Okay, that's correct. So what this suggests is there's an energy transfer from hepatopancreas, which is an energy storage location. It's a pretty important energy storage location. That's moving to the gonads, indicating that there's gonadal growth going, and that energy is being used to support it. So clearly these males are ramping up for reproduction. So this is probably a normal thing. Okay. Now we can look at the same data for females. So the female data is a little bit interesting because what you see here is that they divide ovarian development into 3 stages. Stage 1, stage 2, and stage 3. And in stage 1 and 2, you actually have a positive slope between these variables. And what this indicates is that this is probably a period of energy accumulation. The shrimp are storing energy in order to prepare for a bigger reproductive event down the road. And this is what stage 3 suggests. So you see, we go back to this negative slope here that we saw for the males. So likely there is something going on in stage 3, this later period of ovarian growth, that requires a major investment of energy. So at this point, when they've stored all this energy in stages 1 and 2 in the hepatopancreas, now they're transferring all that to the ovaries, probably in preparation for a reproductive event. Okay. And so this is just an example of how you can use the relationship between these 2 measurements, or the measurement of hepatopancreas and anything else, to try to get an idea as to, you know, how they're utilizing energy at any given point in time. Does that make sense? Any questions? Okay. So things that are indicative of energy use. So lipid content of tissues is one of the ways that we can look at energy storage, because there's a lot of energy stored in lipids. Probably one of the historic and simplest ways to assess how much liver is stored in tissue is to do a solvent extraction. In fact, there was a point in time when that was the only way to assess the fat content of any type of a tissue. And I have done these. I have materials to do these. But they don't use this as much anymore as they used to, particularly for measuring whole body fat, because you actually have to kill and chop up the organism in order to do it. But it does work. And so in this particular case right here, mitten crabs again, same species of the picture I showed you of the pancreas, they were looking at the lipid content of the hepatopancreas during the molt period. And so, as you know, all arthropods, they have to molt because they have external skeletons. It's the only way that they can grow. So what they found was in the pre-molt stages, which is C through D4 here, what you see is that the total lipid, okay, the total lipids are increasing rather rapidly here through these stages, all right? And then when we get to the final stage here, the actual molt stage right here, post-molt stage, you'll see that the lipid content of the hepatopancreas drops like a rock. What's the interpretation of this? Go ahead.
How are lipids related to fat again?
They are fat.
Huh.
So lipid and fat, they mean the same thing. So they're, what they're measuring here is a lot of triglycerides. So what's the interpretation of this graph? Pre-molt, post-molt.
A lot of those like stored lipids go towards producing the new,
Going towards the what?
go towards producing the new shell.
Why not? You would figure that molt's going to require quite a bit of energy. So these pre-molt stages here represents a period of energy storage. In this particular case, the acquisition of stored energy is lipid. And then when molt occurs, they just spend all that capital, just like going to the bank. Okay? So this is a slightly different way of looking at the energy content of the hepatopancreas compared to what we looked at previously. Okay. So lipid accumulation during pre-molt was due to the increase mostly in neutral lipids, primarily triglycerides, suggesting energy storage. Okay. So you should be getting a pretty good feel right now for just how important the hepatopancreas is to energetically funding any sort of growth, reproductive events, a lot of important things that go on inside of its body. Now, another way of looking at how much energy is in a tissue is through the process of bomb calorimetry. Any of you ever been exposed to bomb calorimetry? Well, heck, let's do that then. So bomb calorimetry is a method where you take some sort of an organic substance, all right, and you mash it into this little pellet, and you stick it into this little device, and you make it really hot. You literally burn it as much as you can burn it. And by doing so, when you combust it, it will release heat. And the amount of heat that's released is going to be equivalent to the energy content of the dry matter that is contained within the tissue that you are testing. So, I mean, if you make a little pellet out of 10 grams of stuff and you burn it up, it's going to produce an amount of heat that's equivalent to the total energy stored in that 10 grams of stuff. Okay. Now, the problem with bomb calorimetry is it's a very time-consuming method. You got to imagine, you got to collect it, you've got to dry it, you've got to weigh it, you've got to squish it into these little tablets, you put it in this thing, and you fire it up and you go. And if you've got this big experiment going and say you've got a sample size of 200 things, then the amount of time invested in just measuring the energy content of that that's a lot. And so some people get around that by developing models that will allow you to accurately estimate the energy content of tissues based on some variable. In this particular case, what they did is they plotted the energy content based on bomb calorimetry as a function of the moisture content of the tissue that they put in there. In this particular case, it was a pet pancreas. And they did that for 2 species here. And what they found was that there was a tight correlation between water content and the energy content of the pancreas. And you can see how tight the lines are here. And so what this allows them to do is to forego this real time-consuming method. And as long as you know what the moisture content is, and you get this by drying it out, which you would have to do for bone calorimetry, and the moisture content is going to be related to the mass of however much pancreas you were using. And so you can use this regression model here to estimate how much energy was in that pancreas. Okay. So, so basically a large hepatopancreas is going to have more moisture in it, total moisture in it, than a small hepatopancreas will. And so this would allow them to make their estimates much more quickly if they had to do long calorimetry. Now, the authors, I don't think the authors use this method, but I just thought I would introduce it to you because it's another way of measuring the energy content of something. Okay, any questions about energy content of the hepatopancreas? Yes.
Can you actually go back one slide? I'm kind of confused. So you said that the large hepatopancreas would have a higher moisture content. Why is the slope decreasing then? Like, why would you have less energy if it's larger?
Well, no, actually what's plotted here is percent moisture. So what this is saying is that when you get an amount of stuff, if it has more water in it, it'll have less energy because there's less organic material.
Oh, okay.
Answer your question?
Yeah.
Okay. So let's switch gears a little bit and talk about the immune response of invertebrates. So we had a case study where we looked at respiratory pigments. Okay. And that's part of the hemolymph. But it's not the only part of the hemolymph, the invertebrate blood. Invertebrates will also have these cells that are called hemocytes. And the best way to think about hemocytes are as the white blood cell equivalent of the invertebrate, because they play a role in the immune response. And specifically, they're phagocytic cells. They go around and eat things up. And so if an invertebrate gets infected by something, then these hemocytes will be activated and mobilized to go and try to clean up the infection. Like I said, very much like a white blood cell. And I just threw these up here because I thought that was better than white space. But these are pictures of, these are actual pictures of hemocytes. Okay. There are a whole bunch of different types of hemocytes, and we're not going to go through all the different types of hemocytes. If you really want to memorize them for no good reason, I'd be happy to provide you with a link. All right, so in this particular example, what we have, this is actually from larval Drosophila, is the immune system activation in response to parasite load that is coming from a particular type of a wasp. And what they did is they measured this by literally counting the amount of hemocytes that was in their hemolymph. So they measured this by flow cytometry, which is a method that you use to count cells. I don't know if we do that here. Anybody had a cell class if we've done flow cytometry?
We can for our experiment if we want to, but we don't do it in the lab unless we ask.
Okay. Yeah, I didn't remember if we had a flow cytometer or not. I thought we did. But anyway, it counts cells is what it does. And so here we have the Drosophila larva. They're infected by a parasitoid wasp. And what you see here is basically our control up here. I know I've got my little picture up there, it's kind of hiding it, but you have these black and these little green ones up there, so it's pretty normal. But when you look down across, particularly these two middle ones, you see a pretty strong upregulation of two varieties of hematocytes. These activated I'll say the word. Plasmatocytes, thank you. And then these lamellocytes over here. So they are represented by the kind of orange and green, or orange and red, pardon me. And you see that they substantially step up post-infection, and there's a timeline on infection. You can see it along the top here in terms of a number of hours. And so what this is illustrating is that the immune system of these invertebrates, in this case a larval invertebrate, really step up in response to an infection. And so you can use a measurement of hematocyte activation as a way to test whether or not the immune system is being upregulated in the invertebrate. Does that make sense? All right. So here's when you start looking for an article to present your lightning talk on. Goodness, I may need to, because I think we may need to start our lightning talks actually on Wednesday. Whitney's really sad eyes. All right, we'll still start them on Monday. I have enough to talk about on Wednesday. You're really good at that, you know? Okay. Anyway, I'm not going to hold you here for 4 minutes to do that, but your task, and maybe what we'll do on Wednesday is if I finish the rest of this case study, I'll give you the rest of Wednesday in here to work on your lightning talk. So you need to get some ahead of it. Are we all agreeable to that? Although we should send Ian an email and tell him that he's getting his on. All right. Everybody have a great rest of your day.
Thanks, John.
You're welcome. I hope everybody's excited about parasitic infections of crabs.
Always. Yeah. Sometimes I'm like, I need more
Our next case study is really cool. It's energy storage and flying bugs. I've looked on the calendar. It's before last. Probably. But not looking at my calendar, I can't say for certain. Well, can you send me an email? Yeah, just send me an email, and then and like I say, most likely I will. Oh, okay. You're welcome. And I'm seeing you guys at 3 today. Is that today? Okay. Yeah.